Hans Christian Gram, was the brilliant inventor of the Gram staining method. He was a Danish bacteriologist who made a significant breakthrough in the world of microbiology. When it happened, Gram developed this technique way back in 1884 while he was working diligently in Berlin, Germany. Hashtag of discovery. Hans Christian Gram's journey began when he was studying bacteria. His focus was on observing bacteria in lung tissue from patients who had died of pneumonia. Gram was curious and keen to make bacteria more visible under the microscope. He experimented with various stains and methods to differentiate bacterial types. Through his research, Gram uncovered a fascinating phenomenon. He noticed that some bacteria retained a violet stain even after washing with alcohol. Other bacteria did not keep the violet stain, but could be stained again with a different dye. This difference was because of the structure of the bacterial cell wall. This discovery laid the foundation for the Gram staining method we know today. An interesting fact, Gram himself mentioned that his staining method might not be perfect and should be applied cautiously. However, it eventually became one of the most crucial techniques in microbiology. Hashtag explanation of Gram staining. Gram staining is a laboratory method used to classify bacteria into two major categories: Gram-positive and Gram-negative. This is based on the structure of their cell walls. Steps involved in Gram staining. 1. Crystal violet stain. The first step is to stain the bacteria slide with crystal violet, a purple dye. Initially, all bacteria take on the purple color. 2. Iodine solution. An iodine solution is added to form a crystal violet iodine complex inside the cells to keep the dye fixed. 3. Alcohol/acetone decolorization. Here is where differentiation happens. Gram-positive bacteria retain the purple color. Gram-negative bacteria lose the purple color and become colorless. 4. Safranine counterstain. Finally, a pink dye called safranine is applied. Gram-negative bacteria absorb the pink/red stain. Gram-positive bacteria remain purple as they don't absorb safranine owing to their already stained state. What do the results show? Gram-positive bacteria appear purple/violet. Gram-negative bacteria show up as pink/red. Importance of Gram staining. Gram staining is important because it helps identify the type of bacteria, guides decisions on antibiotic treatments, provides a quick, cost-effective diagnostic tool to save time and resources.